Hello, my name is David Gitchell, and today we'll be going over what gives proteins its unique traits, its structure. So, first off, proteins are a polymer, meaning that they are a big molecule or a macromolecule. So that means that they're made up of smaller molecules, and these for the protein are called amino acids. Alright, so before we discuss the shapes and structures of protein, we'll discuss one key element of how these structures even start off. So, as we see here in the picture below, there's two molecules on top and a bigger one below. The two molecules on top are the monomers of the polymer protein. And we see here that there's the blue highlighted part, OH and H, or H2O. So in order for these two molecules to come together, form the bottom one below, they must go through the dehydration synthesis. And in our body, it happens in the ribosome in our cell, acts as a catalyst to make this dehydration synthesis happen, to make the macromolecule or the bigger molecule below. And this bond that makes these two molecules come together is called a peptide bond. We see here in the bottom of all these molecules there's also an R. This means the R group, or it's a side chain. Later these side chains will be explained to show how they come together, or how they are the main factor of these molecules or proteins coming together to make certain shapes and structures. Okay, so now we can move on to the primary structure. Now the primary structure is basically the ordering of the single chain of amino acids. As you see here, there's different colorings of the two letters AA standing for amino acid, and the dash between them standing for the peptide bond between them. And so as stated before, the reason why the color code is because it's to show how the amino acids are arranging in order of their different R group or side chain. And together, all these peptide bonded amino acids are called a polypeptide. And below that, you can see the picture, which is just a long string of amino acid chains held together by the various peptide bonds and amino acids. Okay, so the next structure will be the secondary structure. And this is when the single chain shown previously will start to come together through the R groups or side chains. And the two main structures that come with this secondary or second phase is alpha helices and beta pleated sheets. You see the alpha helix kind of has a spiral to it, and the beta pleated sheet is kind of like two strands held together side by side. Okay, so now we'll be halfway through the third shape or structure called the tertiary structure.
I'm like 3 minutes.
And basically, this is where the alpha helix and beta pleated sheet from the secondary structure come together to make one 3D shape for that single part of the protein. As we see here in this picture, there's some alpha helix on the outside, some beta pleated sheets in the middle. Okay, so the final structure will be the quaternary structure. And this features multiple tertiary structures coming together to make one bigger one. As you see here, they're color-coded. If you kind of look into the corners, you can see the beta pleated sheets, and it's obvious the multiple alpha helixes as well. But notice how they all come together to form the final shape of the protein. Okay, since we have discussed alpha helixes and beta pleated sheets, we can now talk about how these sheets are formed. And they are formed through hydrogen bonding. As you see here in the picture in the top left, we have a simple diagram of two H2O or water molecules. So as you see here that there is a bunch of positive and negative signs by these two, by the atoms within these molecules. So the negative signs by the oxygen means that the oxygen is electronegative. So that just means that it's kind of sucking away or hogging the electrons away from the hydrogen atoms. So this is why oxygen would be considered more, negative or electronegative. So similar to how magnets, opposites attract.
The ones with the three pictures that come in.
Hydrogen part of one water molecule attracting to the other water molecule through their negative oxygen. So this is simply a hydrogen bond. So now bringing up the pictures of the two structures, beta pleated sheet and the alpha helix, we can see one in the alpha helix sheet how the red oxygen circles or spheres have these dots connecting to the smaller hydrogen ones. So this is hydrogen bonding with an alpha helix resulting in this specific shape. Same goes for the beta pleated sheet. As you see here, that across the sheet you have more dotted lines that directly say hydrogen bonds connecting, oxygen and hydrogen due to oxygen being electronegative.
Easy, whatever, turn it in.
So one type of effect that creates the tertiary shape, after all the alpha helices and beta pleated sheets have been made, is the hydrophobic effect. And the reason why this creates the tertiary structure shapes is because one, there's non-polar
you can hear your dog barking in the background.
R groups or side chains previously mentioned,
and your father,
or non-polars that don't want to touch the water, and there's polar ones that do want to interact with the water. So with this, the non-polar ones will want to bend inward within the protein, while the polar ones will want to keep on the outside. So this is one of the contributions to a 3D shape within a protein. So another type of bonding is disulfide bonds. So this is specifically towards thiol groups, which has a sulfhydryl base carbon background. As you see here, we have sulfur and hydrogen.
It should be done in 3 minutes.
Once it becomes oxidized, the hydrogen goes away, leaving the sulfur to bond to each other. Thank you for watching this video about discussing the various structures of proteins and how they came to be. Hope you learned something today.
I can hear your dad laughing.
Always, have a nice day.